Effective control of bloodborne pathogens begins with the ability to accurately recognise the potential sources of infection. So what actually are bloodborne pathogens? Bloodborne pathogens are infectious microorganisms in human blood that can cause human disease. These pathogens commonly include the viruses such as hepatitis B, hepatitis C and HIV, and some hemorrhagic viruses such as Ebola, and also bacteria such as methicillin-resistant Staphylococcus aureus or MRSA. A virus is an infectious agent of a relatively small size and simple composition that can only multiply in the living cells of plants, animals or bacteria. The name itself derives from the Latin word meaning slimy liquid or poison. Now, we are surrounded by viruses. In fact, there are more viruses than there are stars in the universe. Bacteria come in many shapes and also many sizes. Some are incredibly useful to us. Think of the ones in your gut. They are amongst the smallest living things on Earth, and they usually consist of a single cell, and they have a circular strand of DNA, a plasmid, that floats in a fluid called cytoplasm. They can cause human disease, and they can produce toxins that can make us really very ill. Although not usually regarded as a bloodborne pathogen, prions should be considered as a proteinaceous infectious particle of note, especially in healthcare settings. Prions can cause infections such as variant CJD, which is related to the heavily publicised mad cow disease. It is a fundamental infection control principle to treat all blood and all bodily fluids, particularly in healthcare settings, as potentially infected with a bloodborne pathogen.